One of the major ways that enzymes differ from non-biological catalysts like metals is in the regulation of biological catalysts by cells. This means that biological catalysts, or enzymes, are regulated by cells. Some methods that organisms can use to regulate enzyme activity are to produce the enzyme only when the substrate is present. This makes sense because organisms want to conserve energy. If a cell runs out of chemical energy, it's going to die. Therefore, many mechanisms exist to conserve cellular energy. It's a big waste of energy for an organism to produce an enzyme if the substrate isn't even available.
One type of enzyme regulation involves enzymes that have more than a single binding site. These enzymes are called allosteric enzymes, and they have active sites that can be altered by the binding of small molecules called effector molecules. There are two types of allosterism. Positive allosterism, this is where the effector binding molecule converts the active site to an active configuration. We also have negative allosterism, and this is where the effector binding molecule converts the active site to an inactive configuration. So if we start by looking at positive allosterism, you can see that we have our big purple enzyme here, and you can see that the active site is unavailable for the substrate. You can see that the shape or the topography of the substrate doesn't match that of the surface of the enzyme. But what happens is there's an allosteric site in the molecule, and this is where the effector molecule can come in and bind. And you see how the effector molecule is going to bind in that allosteric site. But what it does is that it changes the conformation of that active site. So it changes the activity site so that the enzyme becomes a better catalyst and the rate accelerates. Negative allosterism is essentially the opposite. So you can see here that in our big blue enzyme, we have the active site, which is in the correct conformation for the substrate to bind. But what happens is we have an allosteric site, and this pentagon is a negative allosteric regulator. So this is our effector molecule, and when it binds, it changes the activity site so that the enzyme becomes a less effective catalyst and the rate slows down. This is the example of negative allosterism that's shown in our textbook. It says here that effector molecules change the activity of an enzyme by binding at a second site. So again, this example that's shown on this slide is a specific example of negative allosterism. So here's the effector binding site. So you can see it's got a certain shape to that pocket. And then we have our enzyme, which is made up of several protein chains, and there are different colors here, 1, 2, 3, 4. And then we have the substrate, which is bound to the enzyme. But what happens is, after the enzymatic process is finished, the substrate goes through the transition state and gets released as products. But what happens is, you get two different products, and one of those products actually happens to fit just right into that effector binding site. And when it does, when it binds in here, it changes the conformation of the active site. So the binding of the effector in the effector binding site causes a conformational shift of the enzyme that closes or shuts the active site and therefore inactivates the enzyme. An example of allosterism is found in glycolysis, which is the first stage of the breakdown of carbohydrates to produce ATP energy for the cell. This pathway must be responsive to the demands of the body. When more energy is required, the reactions of the pathway should occur more quickly, producing more ATP. However, if the energy demand is low, the reaction should slow down. The third reaction in glycolysis is the transfer of a phosphoryl group from an ATP molecule to a molecule of fructose 6-phosphate. This reaction that's shown here is catalyzed by an enzyme called phosphofructokinase. Phosphofructokinase activity is sensitive to both positive and negative allosterism. For instance, when ATP is present in abundance, it is a signal that the body has sufficient energy and the pathway should slow down. ATP is a negative allosteric effector of phosphofructokinase, inhibiting the activity of the enzyme. Conversely, an abundance of AMP, which is adenosine monophosphate, which is a precursor of ATP, is evidence that the body needs to make ATP. When AMP binds to an effector binding site on phosphofructokinase, enzyme activity is increased and it speeds up the reaction and the entire pathway. Thus, AMP, adenosine monophosphate, is a positive allosteric effector of the enzyme. The next section deals with feedback inhibition. And what we're going to see is that allosteric enzymes are the basis for feedback inhibition. Feedback inhibition usually regulates pathways of enzymes involved in the synthesis of a biological molecule. Such a pathway can be shown schematically as follows right here. In this pathway, the starting material A is converted to B by enzyme E1. Enzyme E2 converts B to C, and so on until the final product, F, has been synthesized. If F is no longer needed, it is a waste of cellular energy to continue to produce it. In order to avoid this waste of energy, the cell uses what we call feedback inhibition, in which the product can shut off the entire pathway for its own synthesis. In this example, the product F acts as a negative allosteric effector on one of the early enzymes of the pathway, E1. When F is present in excess, it binds to the effector binding site, causing the active site to close so that it cannot bind to substrate A. So that means that F binds to the allosteric site of E1 and shuts it off so that it can't bind to substrate A anymore. Thus, A is not converted to B, and the entire pathway ceases to operate. The product, F, has turned off all the steps involved in its own synthesis, just like heat produced by a furnace is ultimately responsible for turning off the furnace itself. When the concentration of F drops, it will dissociate from the effector binding site. When this occurs, the enzyme is active once again. Thus, feedback inhibition is an effective metabolic on-off switch. Another means of regulating enzyme activity involves the production of the enzyme in an inactive form that we call a proenzyme. Sometimes we call it a zymogen. The proenzyme is converted by proteolysis, which is nothing more than hydrolysis of the protein, to the active form when it has reached the site of its activity. This strategy is used with enzymes like digestive enzymes that could destroy the cells in which they are made. An example of this strategy is seen with the enzyme pepsin, a proteolytic enzyme that acts in the stomach. The cells that produce pepsin actually produce an inactive proenzyme or zymogen called pepsinogen. Pepsinogen has an additional 42 amino acids. In the presence of stomach acid and previously activated pepsin, those extra 42 amino acids are cleaved off and the proenzyme is transformed into the active enzyme. So to recap, another word for a proenzyme is a zymogen, and they are an inactive form of an enzyme that can be converted into the active form when needed. Whenever you see the suffix inogen, like in pepsinogen, chymotrypsinogen, and trypsinogen, etc., this means that you're dealing with a proenzyme or a zymogen. The figure down here helps demonstrate what a proenzyme is. So here we have our inactive proenzyme or zymogen, and it's synthesized in the cell with these extra amino acid residues. So what happens is those amino acid residues are broken off, and when they're broken off, you end up with the active enzyme. Protein modification is another mechanism that the cell can use to turn an enzyme on or off. This is a process in which a chemical group is covalently added to or removed from the protein. The covalent modification either activates the enzyme or it turns it off. The most common type of protein modification is phosphorylation or dephosphorylation of an enzyme. Typically, the phosphoryl group is added to or removed from the R group of a serine, a tyrosine, or a threonine in the protein chain of the enzyme. Notice that these three amino acids all have a free hydroxyl in their R group, which serves as the site for the addition of that phosphoryl group. The covalent modification of an enzyme's structure is catalyzed by other enzymes. Protein kinases add phosphoryl groups to a target enzyme while phosphatases remove them. For some enzymes, it is the phosphorylated form that is active. For instance, in adipose tissue, phosphorylation activates the enzyme tracylglycerol lipase, an enzyme that breaks triglycerides down to fatty acids and glycerol. Glycogen phosphorylase, an enzyme involved in the breakdown of glycogen, is also activated by the addition of a phosphoryl group. However, for some enzymes, phosphorylation inactivates the enzyme. This is true for glycogen synthase, an enzyme involved in the synthesis of glycogen. When this enzyme is phosphorylated, it becomes inactive. The convenient aspect of this type of regulation is reversibility. An enzyme can be quickly turned on or off in response to environmental or physiological conditions.